We understand the challenges of cross-reactivity and background noise in traditional assays. Codetta's noise-canceling technology addresses these issues, allowing for precise and reliable multiplex analysis. It eliminates background noise in multiplex assays, enabling you to analyze multiple targets simultaneously, without having to titer down your detection antibodies for one target to prevent off-target binding to the other beads. This capability enables researchers to expand their assays without compromising specificity or sensitivity, ensuring high-quality results every single time. Our unique noise-canceling assay design uses fluorescently encoded microbeads and precise PCR amplification. This ensures that only true positive signals are amplified, eliminating background noise and false positives. This innovative approach empowers researchers to perform high-precision multiplex assays with confidence. The noise canceling I mentioned earlier is a function of how we deploy our PCR primers. On the surface of each protein bead, we have immobilized a specific antibody along with a specific forward primer. That primer matches the DNA tag found in the detection antibody for the target, but not any other. Since each target has a detection antibody with a unique tag sequence, productive PCR can only occur when the correct detection antibody is paired with the matching target bead. Off-target, nonspecific binding events can't produce amplicon, thereby turning off the noise that is commonly a problem in multiplex immunoassays. This allows us to reduce background signal and enhance sensitivity while in multiplex. In this figure on the left, the only antigen present in this multiplexed assay is IL-1 beta. However, you can see that without noise canceling, significant false positive signal was generated for IL-1 alpha, and to a lesser degree, the other analytes in the target mix. On the right is a data produced with noise canceling turned on, and the same dose response for IL-1 beta now has a clean response with extremely low background for the other targets. Single antigen dose response is depicted here for each target, showing typical background signal. Here we demonstrate the impact of our noise canceling technology by eliminating nonspecific signal. We enable highly sensitive assays that maintain specificity even in complex multiplex settings. This ensures that you can trust the data from every run, making Codetta a reliable partner for multiomic research. Let's talk about a common problem researchers face, measuring both low and high concentration samples in the same assay. At low concentrations, signal can be too weak for accurate quantification. At high concentrations, signal saturation often limits the precision of measurements. This dual challenge creates significant hurdles in obtaining reliable data across a wide range of sample concentrations. Now here's how Codetta's technology solves this. Our platform uniquely integrates digital and analog quantification onto a seamless process. At low concentrations, we leverage digital quantification techniques similar to digital PCR, focusing on the fraction of positive beads to ensure precision. As concentrations increase, the system transitions to analog quantification using qPCR methods, which accurately measure even when most beads are positive. By combining these approaches, our platform offers a wide dynamic range, ensuring high precision and reliability for both low and high concentration samples. With Codetta, you're no longer limited by the constraints of traditional methods. This innovation gives you the accuracy you need, regardless of concentration levels. Another unique feature of Codetta's technology allows us to quantitatively measure samples in both the low concentration digital range as well as the more concentrated analog range. Let me explain what I mean by that. Consider the bead array when there are a few targets. There will be many negative beads, and the concentration of the sample can be derived from the positive fraction of the beads, similar to digital PCR. As the concentration of the target increases, a great fraction of the beads will become positive, until all or most of the wells will develop signal. At this point, analog quantification is used to determine the targets per reaction. using the cycle threshold as in qPCR. By combining these two modalities, we measure samples over a very wide dynamic range. For example, in this data we have here collected for IL-6 over 6 logs.